So our first keynote speaker for today is Professor Dr. Samlee Mankhetkorn. For his academic position, he is a lecturer at Chiang Mai University. He is also working at a cancer research and treatment center. Professor Dr. Samlee will do a live lecture for the topic of application of liquid biopsy, light cell imaging and plasma proteins, electrophoresis in natural medicine, identify causes and access the treatment outcome. And just a little inform you before we get started. If you have any questions during the presentation, please type them into the chat box. I will bring them up during the presentation and we'll also have time for the questions at the end. Now, we will turn the time over to Professor Dr. Samlee Mankhetkorn. Please give a background of approach to welcome him.
Thank you to the moderators for your nice introduction. Dear all participants who are joining the second International Conference 2022,000. First of all, I would like to express my sincere thanks to the conference organizer and the committees, especially Dr. Kauri, who give me the opportunities to share my knowledge and perspective on the management of the integrative medicine as another evidence-based medical processes. I'm very honored to be invited and to be here to give a keynote speech on application of cell-based liquid biopsy, light cell imaging, and plasma protein electrophoresis in natural medicine. I will focus on the identified causes and the assess the treatment outcome of YAMUR polyphenols. In fact, this is the first demonstration of bridging the gap of natural medicine and the interlockings of cells as a body basic building blocks and microenvironment. It is the reason why the natural medicine should deal with the concept that builds on the extraordinary of body to heal itself. First of all, I would like to introduce you the place where I continue my research work. My place is in Doisket, in Ampheur Doisket, Chiang Mai province. You can see it very nice area, and we have an institute with laboratory here, and also here it is a center of cancer research and treatment center that I deliver my lectures. Let's see the concept of illness and disease and sickness. So the German philosopher George Gadamer wrote that general feelings of person well-being appear mostly when we, in our feeling of personal well-being, are open to new things, are ready to start new business without considering demand made on us. So if we consider, there are three terms to indicate the pathological state: illness, disease, and sickness. From the beginning of human society to the present, a wide variety of medical knowledge has emerged for her happy existence during the life that the nature has determined. As is the case in the prehistoric time, humans do not consider disease to be a part of nature, which was later often assigned to the activity of God. Later, wisdom medicine was born, in particular, Babylonian, Chinese, Egyptian, and Indian medicine has begun to treat disease with plant and natural substances. In the meantime, medical science continued to develop until the 18th century. Western medicine was born. Western medicine has brought medical science to use primarily to create a knowledge by organized research study to observe natural phenomena of the bodies. Then they use the data obtained to synthesize a hypothesis of the coincidence and occurrence of the diseases. Follow-up assessment of hypothesis through the observations. In this perspective of views, the Western medicine is known as evidence-based medicine. However, Western medicine focuses only on detecting lesion of disease and managing the disappearance of those lesions by means of medical science, such as synthetic chemical drugs, surgery, irradiation treatment, and so on. Today, it was recognized that Western medicine is unable to detect degeneration of cellular system before injury of cells, consequently to damaging of tissue, organs, and body. In fact, life is interlocking of cells and their microenvironment. Medical science has been a great advancement in the past decade. In particular, go deeper and deeper in details and clarify the nanometer scale structure of the body. This makes clear the relationship between structure of the body and mind as well. Scientists explore or study the life at the nanoscale can bring proof as indicated in the diagram. And the knowledge fields of life can group as indicated in the pictures. And they also orchestrate together as I showed in the slide. Scientific knowledge study life, and then we propose the ontology models. We can separate the bodies from, we cannot separate the body from the mind, you can see. It can be seen that illness affects the entire organ system of the body, not just single organs. Of course, the interlockings of cells as the body basic building blocks that coexist with the microenvironment allow them that can make appearance the bodies and life can be seen molecular by using molecular imaging, as you can see. So we synthesize the paramagnetic compounds by attaching the mechanism to quercetin molecules, as you can see. The complex is physical level inside the cells, which has intercellular communication in the mitochondria of the cells. This makes it possible to produce MRI images that focus only MRI signal of the mechanism and allowing visualize the distribution of cells in the body, as you can see. So it is a control in the rat, Vista rat. You can see it is a control without any tracking. And after we use the gadolinium DTPA, it is a conventional contrast agent used in medical imaging. We can see more in details, for example, spinal cord, brain. And when we use the cellular tracker like manganese, quercetin, we can see in details, you can see that normally we see the cells. So we can say that the body is composed of cells, and the cell is really the basic building blocks of our body. So we recently developed a direct monitoring of cell function in vivo by direct measurement of the intensities of infrared radiation emitted by the cells due to cell metabolism and cell activities. Then we convert the infrared radiation intensity to the temperature in the degree Celsius unit and mapping the organs in the body. Indeed, it has been transformed into image of tissue and organ showing the distribution of heat in the organ of interest. So this helps us to see the cells in the body system. Let's consider the cell as the body basic building blocks of our body. You can see it is a diagram demonstrated the composition of our body. When we take a cubic of sample from anywhere of our body, it consists of 4 types of tissue, like muscle tissue, nerve tissue, epithelial tissue, and connective tissue. And each tissue composed of cells and non-cells. The non-cells consist of glycoprotein complex. It is the structure of, in fact, you can see that it is only cell is a living system in our body. In order to verify if our body consists of cells and the extracellular matrix to constitute together, so we use a technique that we call liquid-based biopsy. So in fact, how we do? So we use the technique that we correct the blood from the blood vessel, and then we isolate the blood, and we analyze the cell in several conditions, in particular by cell culture, and we use live cell imaging and flow cytometers. So the process of live cell study can be done as follows. So we isolate the cell from tissue. That in this time we use blood as our tissue sample, and we reconstitute the cell mimic to their own microenvironment and control the microenvironment factor like in the body. At the same time, the cell behavior can continuously examine. So let's see, when we correct the blood and we preserve the blood in our conditions, the blood was injected in the tube that coated with heparin, lithium heparin, and we gently mix the blood up and down, you can see, and then we observe the smear of the blood here, you can see, and we observe the color of the blood. For example, in normal condition, we should find reddish color. After we centrifuge the blood, the tube, we found that the red blood cells were sedimented to the bottom of the tube, and we can see really thin buffy coat here and the plasma here. After that, we isolate the cell that found in this area, and we obtain the white pellet here. It is the nucleus cell, including embryonic like stem cell, nucleus cell, and granulocyte. So we try to demonstrate that the blood is connective tissue. You can see that after we correct the blood and we study in a big group, big populations, like 8,000 people, and I just select a representative blood sample. You can see it's very interesting that the blood that we obtain from our study has a variety of difference in both in color. You can see in the content of the red packed cell here and the plasma color here. And the most important thing is, you can see this, the content, the amount of the white pellet that normally consists of embryonic like stem cell, nucleus cells, and the white blood cell also. You can see it. It's vary from very small amount to high abundance. This signifies some conditions in our body, like for example, it is MDS, for example, from MDS group, and from leukemic person here. We found that normally the blood plasma was found about 45 to 55%. After we collect the blood, we separate the plasma and the pellet of the cells. The pellet of the cell was resuspended in a cell culture flask containing the culture medium that specialized or made in our laboratory. The blood plasma was stored at -°C and analyzed using electrophoresis. You can see the live cell that we collect from the blood. After we introduce to the container or the culture flask, and we resuspend them using the culture medium and press in the incubator, mini incubator, press on the stage of microscope, and we observe the behavior of cell, and we can quantify or count the cell number using this system. And we just observe by the idea that we should not disturb the system. You can see this. So after the cell let incubate in the mini incubator, we observe the morphology. You can see it's difference in morphology. So the microscope that we use has high resolution and high magnification enough, so we can identify the cell without staining. You can see when we see or we observe a specific phenomenon, and we would like to identify the communications or how they contact together, we use cell stack imaging, as I showed in the slide. We try also to use the live cell image, the bright field. That means we image the cell using the bright field technique, combined with the fluorescent technique, and try to identify the type of the cell with the different antibodies. You can see. So we can, by this way, we can identify what we see in the bright field, choose corresponding to the specific type of the cell. So let's see what we obtain from the white pellet after we isolate the cell from the healthy donor. You can see that normally the cell that we obtain, you can see they have specific morphology, like a hemispherical shape, you can see, and some of them has irregular shape. You can see this. In the culture flask, we can classify the cell into group. One, they were suspension in the culture medium. Another was go to the bottom of the flask and attach on the flask. After we use high magnification, you can see we observe a clumping of cells here. Very small cells come together, and then we see this. You can see, in fact, it is a cell. They have very small in size. You can see the size of them vary from 3 to 5 micrometers only. After we identify using molecular signature, the cell corresponding to the embryonic-like stem cell, you can see. The cell has many morphology and have specific behavior. We can observe the cell, you can see irregular shape and attached onto the bottom of the flask. After we separate the cell and try to understand what are they are the mesenchymal stem cell that go inside, go to attach on the flask. And we found the nuclear cells. It is the mononuclear cells composed of monocyte, lymphocyte, for example. In this. After we let the culture going on for 24 hours, you can see they differentiate in different kind of morphology, like spindle shape, have different cell morphology, small, and they have some colony like I indicate here. So in fact, the cell that we obtain from the blood has specific characteristics. So they have presence stem cell characteristic, and they have many types of stem cell inside. At least we found they are embryonic-like, that they are very, very early stem cell, early stage of stem cell in our body. And we observe some mesenchymal and hematopoietic stem cells here. And after we let the cell going on to 60 hours, we can observe the cell can originate a complex tissue. You can see this. It is what we observe is the micro. It is neurovascular bundle. You can see this, and the stem cell come together and form a tissue like this. In fact, the cells that we obtain from the blood has very interesting characteristic, like they consist of stem cell, they consist of the cell that come from another tissue, they have the property of regeneration. So this makes us to explore more. How they can do it in this way? Let's go to the theory. So after fertilization, so we obtain the zygote, right? The zygote normally should move from the fallopian tube to the uterus of the mother. So they divided themselves by two, right? At the stage of 16 cells, you can see it is called blastomia. If we take this group of blastomia and separate the cell and perform the cell as a culture, we should obtain 16 blastocysts normally. This signifies that the cell in this stage has totipotency, you can see. After that, we let the cell go on. They come here, you can see. And then we obtain the morular stage here. The cell normally should fall together and form a cluster like this. And then they form the blastocyst and gastrulation, and then they implant in the uterus of the mother. We found that there are a group of cells that found inside the blastocyst that normally we call inner mass. It has a pluripotency characteristic. The cell was assigned to quiescence or found in a speculation lesion in the tissue we call the stem cell niche. So the cell can normally the cell has very small in size that we call normally 3 to 5 micrometers, and they has a specific microenvironment. When they was triggered by the signal from specific area, they should go out from the knees and go to the blood vessel and divide them, entering to the cell cycle and dividing themselves. They divide in what we call asymmetric division. So the small one preserve their characteristic and go back or homing to their origin niche. And the others, big one, they undergo proliferation, and they can go to the lesion, or they can regenerate the tissue. So in fact, the cells we call embryonic-like stem cell was found in all tissue. You can see it is the source of cell that very, very important for repair and regeneration of our body. So in fact, the body has the stem cell that has very, very potency to repair and regenerate our body. After we correct the cells and resuspended or reconstitute them back to the mimic physiological conditions, and we observe the cell under microscope, we should find the cell behave like this. It is a representative study. You can see the cell can move. They change their morphology all the times. You can see. After we try to analyze the behavior of the cell, we can see the cell move. You can see they have what we call the specific trajectory like this. And we found that the velocity of the cell is the movement of the cell has the velocity about micrometer per second. It's quite rapid, you can see. So the cell can go and try to contact, and in fact they interact together, you can see. So in order to demonstrate more of what they do exactly together, we focus on this. You can see that after we suspend the cell in the culture medium, you can see the cell can move, and the velocity of the cell is about, in this case, 20 micrometers per second. Is very rapid. We observe the cell that normally you can see the membrane system is very active. You can see the membrane is quite active. They form a kind of pseudo membrane, you can see, and they send a small vesicle outside, and then they draw back again, you can see, like this. We call the microvesicle. The microvesicle contains the communication proteins that they use for communication at the long distance, you can see. When the cell was contact with the microvesicle, they change the morphology and behavior, while the original one changed their morphology also. In this case, the cell can proliferate and migrate and to form the communications. In this case, they form a tissue. You can see that this video clip demonstrates a communication pathway of the cells. It consists of the microvesicle, here you can see, and they have a cell-cell contact like this, you can see here. And they also create what we call intercellular bridge to connect the cell together in this way. It is the basic, the principal pathway of communication of the cell, you can see. In fact, the cell life become together by communications. They need communications among them. And how they behave when they were found in a real tissue, you can see they move. And all the cells move. We can see the dynamic system, you can see here. And let's see. You can see it is the cell. They can attach on the cellular matrix, and they can leave from the extracellular matrix, and they go to everywhere that they would like to go. While they mediate communications among them every time, you can see. So the cell is a living system that are very active in our body, and they actively communicate among them. The communications among cells is the primordial living communications, I think. Let's see when if the cell were found in damaged conditions, such as they were treated using chemicals for it is a sample from the patient who get retinal detachment. After the treatment, they use a vaccine that for long times. You can see the cell change the morphology. They become like a fibers, and the cell, the healthy cell has very small amount in this before joining our study. After 1 month, we found that the cell, we can recover the stem cell, you can see, and they can generate the neurovascular bundle, and they form the tissue inside. It signifies that when the cells was damaged or was introduced to the damaged conditions, stress conditions, we can recover. And it is the stem cell that come from the niche and go to the blood vessel and perform such kind of activities. In fact, the cell remember the trigger signal from the cells. You can see this also from the stroke person. You can see they got stroke for 10 years. After, when we analyze the cell at the beginning and before treatment, you can see the cell has very low abundance, and they were found in stress conditions like this. After we regenerate the conditions, we observe the stem cell that come and regenerate the neurovascular system. You can see it's very exciting result, you can see. So in our body, in particular in blood, there has the cell that has many kinds of cell, more than blood cell, you can see. So here I would like to mention that the blood should consider like what we call connective tissue. It is not only the blood that we consider only white blood cell, red blood cell, and platelet. In fact, it is the what we call the source of tissue that we can use for investigate the phenomena in the body, because of it is easy to correct, easy to isolate the cells, and of course, because of the blood has the extracellular matrix as a liquid connective tissue, so we can reconstitute the environment for the cell mimic to the physiological conditions. After we let the cell going on in the culture, you can see they can originate a complete tissue. You can see very beautiful, and you can see if you see in this picture, we use live cell staining to stain the mitochondria. You can see the bright inside the cell here, the nuclear compartment, lysosome, and the cytoskeletons that we can demonstrate. After we obtain the cells from the blood, and we can regenerate and many kinds of cell tissue type in the condition of birth. It is very important finding that I would like to communicate with you that we have a chance to obtain a blood sample from a monk, a spiritual monk. You can see. After we collect the cell, we observe that the cell consists in the cell microenvironment. We observe the cancer cells. In fact, he got cancer, colon cancer. So we observe the cancer cells, and the cancer cell can undergo apoptosis. Very impressive for me, because of cancer cells normally they do not respond to apoptotic cell death. After the cancer cell disappear, we found that the cell behave in this way. You can see they mediate organization, and they migrate to become a cluster. You can see this, and then you can see it's really well organized. You can see, and then here we observe that after we observe this condition during I think within 2 hours after we isolate the cell generate this. You can see, and this also you can see. In fact, they become together and form what we call neurovascular bundle. Here you can see it is a kind of a perfect conditions. That means the cell can generate the tissue even in very few hours after isolation. You can see we also observe the tissue cup that long after identification, it is internal mucosa tissue, and we observe the small dark like a vessel here. You can see it is a neurovascular. You can see, and it is the complete tissue. This makes us to hypothesize that the cells that need microenvironment, and they can generate the tissue, and they can compensate the tissue that was destroyed also. You can see. Very interesting. So this signifies that the cell from the blood has many characteristics, and they has the memory system. They can remember what they should do. In the same case, if we observe well in a spiritual person, you can see the cell normally can change the morphology also. We can observe the cell into morphology. You can see it is very specific. When the person has good health, that means the microenvironment, the around the cell become complex and very healthy, and the person get good mind also. The cell change morphology in the pyramid with base square shape like this and dome shape like this. So we observe that after the cell become in these conditions, they can form pictures like this under microscope. You can see we can observe many kinds of pictures. This signifies that the cell has something that we call knowingness, and they have consciousness also. It's really, very interesting, and it is the first time that we observe this also in literature. Let's see how we can observe this. A person who has the pyramid and the dome cells, at the beginning in 3 years ago, they come to join our project. We update with the prostate enlargement. You can see the cell has found in what we call stress conditions. They change their morphology, and they have many macrophages that normally the cell cannot give or cannot go or survive in long-term cultures. After treatment using cyamopolyphenols, we observe that the microenvironment of the cell change, you can see like this. And then we observe a kind of cell, you can see like a square base pyramid, you can see. And then more clearly see, you can see this like this, and very well shaped like this. And after that, we observe in this way. It is a crystal of the pyramid. You can see how they come. In details, we try to observe when the pyramid appears, they can induce another cell to change that morphology to the pyramids also. And we observe many kinds of pyramid cells like this, and this, and this. After that, we observe the environment, in particularly the culture medium, move like a cycle, but they turn light like this, and they bring the pyramid cell to come together and form a kind of group like this, and form a crystal of cell like this. It's very, very impressive result. And the cell do not still living, you know, they did not dead yet. And they can turn left and release from the crystal to become a single cells again. And what they should do? In this time, we observe that the cell can change behavior. They migrate to be together, but this time we observe that they can form a kind of pictures like this. If you know what we call Buddha, right? It looked like a kind of the sculpture of Buddha. You can see this. Very, very interesting. It is found in the laboratory, but not only one sample. We found in many people who practice vipassana meditation, for example. So I would like to conclude the findings here that one cell is one life. The relationship between the spiritual and the cell is the basis of the formation of the body. The mind of the spiritual is a natural way aware of emotion, forms, sounds, smells, and taste. When we consider cell is a big data system. They have memory system and process massive amounts of information quickly and accurately. Cell has knowingness, thus presents the consciousness. The number can be increased according to the system requirement. The cell morphology is fundamentally depends on the function and the responsibilities. And of course, they was controlled by that program. It is the nature. So in order to get further insight, what we observe exactly, we try to explain the conditions. This time we try to analyze the plasma protein. We focus on the secretory proteins that are belonging to the cell-cell communication proteins. It is a big challenge for us because of to handle the plasma protein, the plasma in the lab is very complicated and very high cost because of we need to store them in minus 20 degrees Celsius. As we have many samples, so it's a very big plasma bank. So we develop a system that to preserve the plasma. In particularly, we use lyophilization. So we develop a technique that we lyophilize the plasma in the in the powder, you can see, and then we try to identify the proteins and the activities, and then we what we call create a technique that try to that we can use to consult the result. So we use the web-based application for analysis the protein. So in the lab, we found that at least 42 proteins in the blood plasma that concern to the cell-cell communications. And by using bioinformatics, we can classify the cell, the protein in 8 groups. You can see this, we call node. In fact, the 8th node is very essential and primordial for repair and regeneration. So in fact, we think that we found the diagram that explains the repair and regeneration of tissue in the bodies, as I indicated here. So what we have found during, it is almost 30 years of work. So we found that the cells join together and operate among them as a basic body building blocks accordingly to their knowingness and microenvironment. The human body is composed of approximately 60 billions of trillions cells, where the cell create the system biology, which is strictly controlled by homeostasis. As an individual living organism, cell has memory system that can efficiently and precisely manage the new data. Every stimulus and every response to the stimulus that occur all the time is recorded to the memory system. During the repair and regeneration, the regeneration process of a given injured tissue, stem cell and trigger cell were triggered, and these events of stimuli were recorded in the memory system. After triggering by particulate stimuli, stem cells left from their niche and migrate into the blood vessel, where they entering the cell cycle and proliferate to increase cell number. By using an in-house system of ex vivo monitoring of live cell, it is also the first time that we can demonstrate the nanostructure of the human bodies. The tissue are fundamentally composed of a large number of cells coexisting with the extracellular matrix system which are fibers of glycoprotein compounds. It is synthesized and secreted by the cells itself, and waters, chemicals, energy, and many other factors that coexisting and fit together with cell and cannot be separated. Otherwise, life will not go on. In this condition, we call perfect state that allows cells to interact with their microenvironment around themselves and communicate with surrounding cells through three fundamental processes: cell-cell contact, intercellular bridge, and microvesicle. Cells and their surrounding are thus organized tissue, organs, and ultimately a complete body. So, as the cells is only living organism in our body, if it was found to have abnormality at the biochemical and physiological levels, that not leading to several deleterious nor death, this will cause a discomfortable feeling of an individual, even though when the cells were in stress condition for long time and finally re-entering to degenerative conditions, they will lose communications among them, resulting in a degeneration of tissue and body. This comfortable feeling and degeneration is such the symptom, which is lacking of any lesion or scarring of tissue that cannot measure by medical scientific parameters. That it is not condition of illness according to the Western medicine concept. Furthermore, direct observation of cell behavior, the secret omics, is play important role in identifying the cellular communication and response to their microenvironment. Both first dimension gel and 2D gels, electrophoresis of plasma protein, and whole cell proteomics can provide an additional information of cell behavior and in nanostructure level of the body. The blueprint of plasma protein gets well along with the cell behavior, demonstrating that in perfect situation, there is at least 8 nodes of activity by which the cell orchestrated among them in order to maintain the good health of the system biology. The node of activities are including the INVHA, or we call cell rest, you can see, or the cytotoxic cell T-cell differentiation, and cell-cell signaling, angiogenesis, cellular metabolism, angiogenesis, nervous development, and angiogenesis, and exon guidance. So in fact, the basic structure that we should observe in the system is the neurovascular bundle. It is what we call the basic structure before complete tissue construction. So we, as I mentioned, now how society have changed in their approach to illness, and this is from ancient time to the present. As we compare to the ontology models at the beginning, which aim at eliminating the system, the functional relational models consider the illness as a dynamic events, endogenous reaction to break of a balance. Right now, in this perspective, the body and mind are inseparable. It is the entire organism that become ill, not single organs. In this model, the physician-patient's relationship is crucial, and the physician promotes self-healing, and the body promote the self-healing process. So we propose the block diagram here. So in normal conditions, when the life were found in perfect conditions, we should observe the specific criteria in the cell count in the cell system like this. The cell should well defined in the morphology. We should observe the immunity system, endogenous system. The cell should mediate cell-cell communication in the adequate manner, well organized to reach an equilibrium state, and demonstrate the tissue regeneration process, what we call perfect conditions. And when the system moves from perfect conditions to the straight conditions, we can see this. In fact, this phenomena is a reversible process, so they can go and back. But in our life, sometimes they cannot go back to the origin conditions due to a limiting step. We identify that the limiting step that found in the condition of health is microenvironments and spirituals. So the cell in the body normally they has what we call the characteristic, like they are big data system, they has knowingness, and thus they present consciousness. They has specific morphology due to their function and responsibility, and they has a very important thing is they control by apoptotic cell death program. So when we found this, how we can bridge the gap of ex vivo live cell imaging at institute conditions to the body conditions. So we try to use what we call we use the infrared imaging, because of the infrared imaging is a kind of imaging that use the origin of the energy from metabolism of the cells. So in fact, we try to identify the cell function in vivo using a non-invasive technique. It is the fundamental of infrared thermography, you can see. So we detect the infrared emitted from an object, and then we transform the infrared energy to the temperature, and then we do a mapping of the infrared imagings with photography, you can see. So it is the IR thermal imaging is made by measuring infrared radiation from the body and convert the measured heat intensity to photographs showing anatomies of the body. So by the way, we can localize the function of cell that we found in the ex vivo system to the in vivo system, as we mentioned here. And then we try to do a translational research from bench to natural medicine, natural medical services by in cooperation with SCL medical clinics here in Chiang Mai. So right now we have almost 8,200 people who joined to the program, and we use what we call the cell-based liquid biopsy, light cell imaging, infrared ray imaging, and electrophoresis for identifying cause and assess the outcome treatment of cyanopolyphenols. So before using in this objective, we produce the Siamo polyphenols in our own laboratory by this system, and we do quality control, and we identify the chemical profile inside, as you can see. So we published many papers concerning the Siamo polyphenols in varieties of journals. We focus on the antioxidant, anti-cancer, and immunomodulators, and endogenous stem cell regulators. So the Siamo polyphenol is a group of a group of polyphenols, and we very well study their physical chemical properties and their mode of transport in the cells. The intracellular target of the molecules were found in mitochondria, so that they react on the mitochondria of cancer cells or stress cell and degenerate transform cell that not respond to the apoptotic program. So they can induce the apoptosis via the mitochondrial pathway, you can see. It is the chemical structure that we study. So let's see, and what we call a sample that we study here. So we use the infrared imaging to identify the change of tissue. For example, here is a person who got cancer in the lung, very progressed cancer, you can see here. So the infrared imaging also identify the region here also, you can see. So in fact, the infrared imaging is a kind of imaging that's very high sensitive and give information about the function of cells in the body. It is an advantage compared with the CT. You can see CT gives a very precise area of the tumor, but they cannot inform you about the live cell or the functions of cell. The disadvantage of the infrared is the area or the shape of the lesion is not clear, but they can give information on the function of the cells. So it is a kind of cell imaging. You can see after we found, we identify the lesions in the body, we isolate the cell from the blood, and we observe the cancer cells also inside. And after we observe the cell, we continue culture the cells for long times and identify the growth curve, and using what we call the biomarker for identifying the cancer cell using flow cytometry. It is an example it is an example that we got a patients, a female patient who got breast cancer. You can see at the beginning she has 2.3 to 1.4 centimeters of the tumor size at the beginning, but she did not follow with the medical centers. So she come to 5 years later, she come to our center, and using what we call the imaging, the infrared technique, we identify almost 5 centimeters of the tumor found in the left breast here, you can see. And after 3 months, after 10 months of treatment, we found that the tumor size decreased. You can see it become like this. When we observe the cell, you can see at the beginning we observed many epithelium cells were found in the cell populations. This signifies that her body tried to regenerate and repair the lesion, but they cannot. So they found in the blood like this. And we observed the cell with they completely changed their morphology. You can see this. It signifies cancer cells. You can see. After treatment, we found that the epithelium cell was lesser compared with the previous one, and we found the T cells, the monocytes, and the cell become a group like this. You can see this, and we also observe some what we call natural killer cells. You can see, and for treatment, the fourth round of treatment, you can see, we observe many natural killer cells. You can see this. And we also observe the endogenous stem cell. In particularly, we observe the embryonic-like stem cell. You can see there, and many natural killer cells inside. You can see. And the fifth round of treatment, we observe. You can see many high abundance of natural killer cells. You can see. So it is very interesting that you can see when we observe the high abundance of NK cells, the tumor size dramatically decreased. You can see from year to year. It is very surprising. So in fact, we should say, consider that in our body, there has what we call archives of self-healing. They try to maintain their health status. So when the system would let or bring back to the perfect conditions, all the cells who normally protect the body go out and try to eliminate the bad structure of the body. It's very nice. It is the similar for, you can see it is a tumor here. It is in what we call in the next here. You can see it is disappear also after treatment of 3 months of treatment. You can see. So you can see also the cell can give more information. So you can see after they receive Siamopolis phenol for 3 months, we observe the cell change morphology. We observe what we call the lymphocyte, in particularly the T cells and the monocytes, and what we call the embryonic like here. And of course, in this person, we observe many natural killer cells also. I would like to show you the infrared ray imaging is very powerful techniques that, for example, we can perform what we call perfusion technique and can identify the tumor found in head. You can see this area. So it is before treatment. After Siamoa treatment, you can see we can see the change of the tumor. So it is very good get along with the cells, living cell imaging, and also the what we call the protein profiling also. One case is very interesting is for a diabetic person. You can see she got inflammation with the trigeminal nerve, so she got Bell palsy, and also she has noisy in the ears. After we observe this, you can see after treatment with polyphenol, we observe neurovascular bundle and also the neurofiber like this. We observe the Bell palsy disappear and also the noisy disappear also. So it is a new thing that can give us information. A neurogenesis exists, you can see. So when we focus on cells, many things that normally we believe that it cannot replace when this damages. So in fact, our body has a system that can regenerate the damaged tissue. You can see this very nice. And also, this is one of interesting results from our research. So a person who got a trauma, he got was accident with a cerebrovascular accident also. You can see at the beginning, he found this stage. After their parents tried to do exercise and massage, Thai massage, and in combination with our system. So right now he become like this. Very, very, very helpful and very interesting. So it is all what I would like to share with you. If you have any questions, you can ask me or you can send the questions to me and I try to respond to your questions. Thank you.
Thank you, Professor Dr. Samlee.